Yeah, I'm working on advancing functional genomics with Bayesian modeling. I've been working on it for 6 years, and it's actually my first time to present this to a Bayesian audience, so I'm quite excited about what you think about this work. Before I start, who of you knows the name of this Tyrannosaurus Rex?
I do the name, but I know where the foot was taken.
But who of you knows the name?
Fragile.
No. So, if you're wondering what to do, go to the Natural History Museum and meet Stan.
Where is it?
Just here in Oxford.
Oh.
It's like six minutes walking from here, so
It's
yeah.
2 minutes from where the football is taking place.
I know it, yeah.
Yeah, so and entry is for free, so I can recommend it. Okay, yeah, but let's get to what I wanted to talk about today. Yeah, so I will give you a short introduction into the field that I work in. Then I will talk about the model development process that I've been going through for my projects. And I will present two case studies, just talk a little bit about where I've actually used this and summarize. So usually I give an introduction to Bayesian statistics when I talk about this project, but I guess today I should rather give an introduction to functional genomics. So functional genomics measures the contribution of genes to cellular processes. It provides insights into biological systems and disease development. The experiments are very complex and have a lot of confounding factors, and they're also expensive, so you usually have few replicates. But you have thousands of genes, so a lot of parallel measurements, which makes it really a very good field to apply hierarchical Bayesian models. So the data that you typically get, you have the genome, and the genome is transcribed, and you have RNA. So this RNA is being cut into pieces, and that's what's being sequenced. And then you do this for 2 different experimental conditions, for example, which could be 2 different, to some disease-relevant conditions, for example. And then you measure the differences between these, the counts that you get for the different conditions. So that's a fairly simple problem. So the effect size is the log-fold change. So what I will talk you through for the model developing process is a much simpler model than what I used in the projects afterwards, which is really just this simple problem. So you have 2 different conditions. The effect size is the log-fold change between these 2 conditions. And I'm trying to optimize this very simple model to then have a model that I can generalize for more complicated problems. Yes, so that's basically what the model looks like. The distribution that's suitable for this is a negative binomial distribution, because you have basically over-dispersed count data. So you have the biological coefficient of variation as a parameter, and I impose hierarchical priors on quite a few of the parameters. So on the dispersion parameter, that leads to shrinkage of the dispersion. On the normalized gene expression alpha, and on the normalization factor between the samples, and maybe also on the effect size. We'll see this in what I will talk about now. So the prior on the effect size, that's something that you can think about a little bit. So basically, one possibility is to just use a broad normal prior, or you can also use regularizing priors, for example, a hierarchical normal, a hierarchical Cauchy, or a regularized horseshoe prior. What's actually interesting is not to look at it on this scale, but to look at it on a log scale, because here you can see what happens in the tail region. And you can see that the regularized, that the hierarchical normal prior has very low probabilities in the tail region, and that will shrink your effect sizes. So let's see what this looks like. So basically, all of those priors perform fairly similarly when it comes to looking at the ROC curve. So this is using data that I simulated from the posterior distribution such that I know where the effect size is supposed to be positive or negative. And when I look at the type M error, I can also see that basically for the significant ones, so that's the dark ones, it's usually around 1 where you would want it to be. But for the, for the not significant ones, you can see quite a difference between these. So I put in here EDGAR, which is a standard analysis tool used in the field. So you can kind of all compare to this. And you can see that the normal distribution looks the broad normal prior looks very similar to what you would get with EDGAR. But two of them have actually a very nice feature, which is a hierarchical Cauchy and a horseshoe prior, that they shrink the effect sizes of those which are not significant to 0, which is something that's very, that also makes the sampling process efficient. Yes, so that's basically what I've said. But in general, basically all priors except for the hierarchical normal prior worked well. So yes, then comes the next challenge. So I have a model that works. The next part will be with the broad normal prior because that's the one that I used first. And you have this nice working model, and then the wet lab person comes and tells you, yeah, but so what do I learn from this? I want to go to the lab and test, so what should I test? So he wants basically to know false discovery rates, cutoffs, etc. And I've really tried and done a lot of reading on this and tried to see whether people have published useful stuff on this, and in my opinion there was far too little on it, and I haven't found a good summary or so, I've developed a lot of stuff myself for this. So the question is, what is it that I want of a hypothesis testing method, right? I mean, first of all, I want to order the genes by relevance. I want that the fitted effect size is similar to the ground truth, so that I want to have the type M error close to 1. I want to identify a cutoff so that I can actually tell people what to test in the lab. And I want to correct for multiple hypothesis testing because here I'm testing 5,000 genes at the same time. So we'll probably always find something. And then it would be also nice if it's a Bayesian concept. So if it accounts for prior, if it uses model comparison techniques or regularization. So let's see. One possibility is to use Bayesian p-values. And here I also use basically an interval around 0 that, well, I mean, I have some ideas of how to determine this interval properly, basically by, I treat it as some kind of, since someone mentioned it before in one of the talks, as some kind of resolution interval. I try to estimate the resolution of the experimental data. So what's like the smallest error that I can find in the data, because below that I can clearly not find anything. Then I get this distribution of p-values. I kind of try to estimate what kind of distribution I would get under the null hypothesis. So you can see that whatever else is basically signal in a sense. The pro is it's easy to explain to a frequentist, but yeah, it's a bit difficult and tricky to underestimate this kind of distribution under the null hypothesis. Another possibility is to use z-scores. Then the null hypothesis is that it's a standard normal distribution, so that fits pretty well. And by looking at the fraction above and below, I can actually get an estimate for the false discovery rate. So that's very nice. It's still a concept that's easy to explain to a frequentist. I have an estimate for the null hypothesis. It corrects for multiple hypothesis testing, but it's not a very Bayesian concept. So yeah, let's see, Bayes factors sounds more Bayesian. So it actually takes the prior probabilities into account. And the distribution looks something like this. There are tables where I can look up what I can actually consider significant for that. Yeah, so it's nice, it's a more Bayesian concept. Tables to interpret them are available, but there's no concept for multiple hypothesis testing here, so I gave up on that one too. Another possibility is to use leave one out cross-validation. yeah, I tried that one too. I basically said, okay, if I, all my findings in this case, I want that model 1 is where half an effect is better than model 0 where I have no effect. So I basically fitted a curve to the left-hand side and projected that to the right-hand side, and I said everything above that curve is basically the stuff that I want to find. So basically, again, by looking at the fraction that's above and below the curve, I can estimate a false discovery rate. It's a more Bayesian concept, but again, yeah, it's a bit tricky where to place the cutoff. Then the last one that I want to talk about is the regularized horseshoe prior. This is a very nice concept because it basically shrinks the effect sizes of those which are not significant or 0 simply, and then you don't need a cutoff on false discovery rate anymore, but you only need a cutoff on your effect size. You can directly place a cutoff on the effect size that you expect to be able to see in the lab. So effect sizes are ordered by significance, and you select the relevant effect size instead of false discovery rate. I'm not sure I can convince wet lab people to give up on false discovery rate, but in my opinion, this should work. So now, how do these different methods perform? So I can see that they actually all perform fairly similarly, except for the leave-one-out cross-validation, which actually had the poorest performance. And also, when it comes to M value, they all perform fairly similarly. So basically, I went through all these methods. We can see that basically all these non-Bayesian concepts actually fare fairly well here. So basically, for ordering of genes, it all works fairly well. And of all of them, the horseshoe prior is basically the only one which fulfilled all the criteria, in my opinion. So let's come to my two projects. In the first one, I wanted to basically know the effect of RNA binding proteins on stability, so they can either block them from degradation or they can facilitate degradation. The experimental setup for this is that you have bacteria with and without the RNA binding protein. You add antibiotics to inhibit transcription initiation, and then you take samples over time, you normalize the data, and you fit a stand model. The effect size here is a difference in decay rate or in half-life. And then we used a broad normal prior, and I used Bayesian p-values for hypothesis testing. And then some of the transcripts, they actually decay like this, so that's just a simple linear model. Some of them, they decay like this, so I can fit this with a piecewise linear model. But actually, most of the transcripts, they decay like this, so they go towards some stable baseline value. And this actually affects 80% of the genes, and it's something that previously has not been taken into account. It's crazy. I mean, people just neglected this fact. They try to fit a linear model to this. It's insane. So basically, I fitted the different models to that, and you can see that it makes a huge difference when you fit what I call the log normal model. You get a median half-life in this condition of 1 minute, and otherwise you get 2.5 minutes, and previous publications even went up to 5 minutes for this. So it's a huge difference, and it's a fundamental biological parameter, which is half-life of RNA in bacteria. And this simple model corrected it really by a factor of 3 to 5. We also found a lot of transcripts which were different between the conditions, so we managed to identify a lot of transcripts that were affected by deleting the RNA binding proteins. And if you're interested in this, we also published this. The other project was Shigella. What about find out more about Shigella, which is a gram-negative bacteria. It's human restricted. It's a causative agent of shigellosis. So there are no good animal models available. And it has a virulence plasmid that's very important for entering the cells. It was done in collaboration with a group in Uppsala, which did the organoid infections. And first, we optimized the experiment. So the technique that we used is transposon insertion sequencing. So you basically interrupt genes, and then you can connect genotype to phenotype this way. But the problem is that in these organoid infections, you have an infectional bottleneck. So basically, only a small fraction of the bacteria will actually infect. So the first thing that we needed to do is we had to determine the number of mutants that we could use such that the technical loss was below 50%. Then we determined how many replicates we need to cover the whole genome. We optimized the enrichment conditions not to select for virulence genes, and then I developed a Bayesian model to analyze the final screen. So basically there were still a few challenges, for example that the samples cluster by replicate and not by input-output. There's still technical loss. And I solved this by fitting two different zero-inflated negative binomial models. The first one for normalization and to extract the technical parameters. And then the second one used these technical parameters and determined the effect size, which in this case is a log-fold change. Here I used Z-scores to determine the false discovery rates. And we actually found many of the known virulence factors on the plasmid, but we also found a lot of previously unknown virulence factors on the chromosome. By follow-up experiments, actually discovered the new virulence mechanism. So that was also a very interesting and successful project because from the beginning we worked together with the experimental group and we could help exactly scale or develop the screen such that we could later analyze it. So I hope I've convinced you that Bayesian modeling is useful for functional genomics. Well, it was probably easy part. Because of this basically optimization of experimental and analysis workflow, we managed to really show that bacteria RNA half-life is much shorter than what people previously thought, and we also managed to find a new virulence mechanism in vivo, and this was actually the first study which managed to do such a screen because previously people did not manage to handle this infection bottleneck. Yes, so technical loss is something that's very common in functional genomics and it often varies between samples. So what I'm working on now is to basically generalize these models to apply them to different problems where you have this kind of technical loss. And for this, I also want to explore the Holschuh and the Cauchy prior because it's very useful, especially for sparse data. So yeah, thank you for listening and thank my collaborators. Yeah.
We're a little bit over time, but while our next speaker sets up, we have time for one question.
Yeah.
Seeing if somebody else wanted to ask a question.
Yeah, I didn't see any other hand.
So I actually got a comment, which is I fit models almost exactly like this and found it to be much more efficient to use the latent parameterization of the negative binomial, that is take a Poisson and take a random effect. It's very counterintuitive because it adds like hundreds of thousands of parameters, but HMC scales really well with dimension.
Yeah, of course.
Right, and the geometry of the negative binomial is really complicated. But the question I wanted to ask is why a cutoff? Why I've never understood why people in this world care about p-values, because what happens is you rank things, right? And you hand these over to the experimentalists, and they only have time to evaluate like 5 or 10 of these. So why p-values? Don't you really just care about ranking them?
Yeah, I care about ranking them, but I mean, my idea is that it kind of helps them because the experiments have a certain cost, time cost and financial cost, the follow-up experiments, and basically depending on how high this time and financial cost is, they want to be like more sure or less sure to do something. So basically, they would like to know how many experiments I expect to fail.
And are
right?
these well
So that's
enough to do that? The p-values, well, because all the stuff I saw, like if you look at Efron's book, the p-values are like 10 to the minus 20.
No, I mean
and
the
then
p-values.
You do multiple comparisons, and it's like 10 to the minus 19, and there's 200 of these that come out, way more than the lab can handle. So do you actually have situations where you get like 5 or 10 that are significant and nothing else is?
No, but the thing is that often when you look at it from a biological point of view, you might, like for example, the first 50 might already be known, so you don't want to use the first 50. But then you found something that's, so you find something that's a bit further off, and then you also have many transcripts that come from the same pathway. So even though you find 20 or 30 more, you don't want to test necessarily the first 10 of them because they are somehow functionally connected. So you might actually sometimes want to go quite far, and actually, or you really look through the pathways and you look for something that could be interesting. Like in this case, this was really not one of the top hits, this new mechanism that we found. It was somewhere maybe hit 80 or something like this, and they actually validated it in the lab, and it's a really interesting mechanism, so, yeah.
Thanks. I've asked like a dozen people this question. You're the first person that answered it coherently. So
Right.
that's really
Yeah. Okay, then. Yeah, then I
We have time
secure-
for another question.
Oh, yeah.
You mentioned pathway data, looking at pathway data to kind of draw on your conclusions. Have you considered maybe including some of that pathway information actually in the prior? Is that something that you can do alongside the regularizing priors?
Yeah, I haven't really tried this or thought about it. I'm also sometimes very doubtful of pathway analysis because it's very difficult to curate them correctly, to actually do some statistics with them. You can often do it and kind of use it in a bit of exploratory way, but to actually put it into the model. I think something else that I have been thinking about, it might be more useful is to put operon annotations, because you have genes that are being transcribed together, and they can often behave similarly. So I think it would be at least that would be a more useful starting point. Yeah.
One more question?
Anyone? Yeah.
Maybe what are the runtimes of these models compared to those classical frequent testings? Is that
Yeah, I mean that actually, I mean they don't scale well at all. Even with parallelization, they run. I mean, it's like this simple model runs maybe only like 2-3 hours, so that's not that much of a. But the big projects, they run several days. And I think the worst one that I had was running maybe 3 or 4 weeks. And
How many genes was that?
that was, so I divided the genome into subgenetic windows basically. So I had like 20,000 or so. And that was times 120 samples.
Let's talk afterwards.
So,
We can
yeah.
fix that. I have a version of this that runs much faster than that.
Yeah, okay, cool. That will be good. Right. Yeah. Okay, then
Great, let's thank our speaker.
Yeah.